Hello everyone and welcome to my presentation. My name is Katerina Georgousaki and I'm working as a safety assessor in QACS laboratories. As you all probably know, in the last decades there is a great progress on exploiting the immense chemical diversity available from the abundant microbial world. Considering that most of the world's microbial chemodiversity remains uninvestigated, we are confident that the identification of bioactive compounds from microbial diversity has a huge potential in the field of cosmetics as well. Thus, the present work is focused on the discovery of novel cosmetic agents from the global microbial biodiversity by using only animal-free techniques. Using the in silico concept of reverse pharmacognosy, we have screened around 40,000 natural compounds isolated from microorganisms against several cosmetic targets. The consensus evaluation led to the selection of the top molecules with potential activity, and by screening microbial databases, we identified potential strains that produce the targeted molecules. In total, 196 fungi were selected, and around 1,000 extracts were produced by applying OSMAC approach. The extracts were biologically evaluated against a broad spectrum of cosmetic targets in both cell-free and cell-based assays. Thus, we selected the endomphytic fungi belonging to the genus Cercospora as the most promising for its antioxidant and skin-protecting activity, since it also showed the capacity to activate proteostasis, ensuring anti-aging mechanisms, and the endomphytic fungal strains identified as new species of the genus Comoclathris that showed the most potent skin-whitening activity, which was confirmed when tested in mouse melanocytes. Bioassay-guided purification of the Comoclathris strains led to the isolation and identification of one novel furan derivative, namely Comoclathrin, and one new sorbicillin analog that demonstrate a remarkable tyrosinase inhibitory activity. More specifically, Comoclathrin shows an around 90 times higher inhibitory effect when compared to the well-known cosmetic whitening agent kojic acid. Preliminary toxicological evaluation of the active extract and compounds showed no cytotoxic effect against a panel of cancer cell lines as well against normal human fibroblasts. The safety of Comoclathrin to be used as a whitening cosmetic agent was further supported by using in silico computational tools such as QSAR Toolbox by ECA that demonstrated the lack of skin sensitization or irritation potential. This was not the case for the new sorbicillin analog. We further focused our research on Cercospora sp extract. Bioassay-guided purification of the active compounds led to the isolation and identification of fulvic acid, which according to the COSINC database has a skin conditioning function. Wanting to further support the use of Cercospora sp extract as a potential skin conditioning agent in cosmetics, we optimized scale-up production in a 30-liter bioreactor by using only non-animal origin ingredients and by enhancing the production of fulvic acid. The extract obtained from the bioreactor showed no cytotoxicity against cancer cell lines, while it was classified as a non-irritant when tested in vitro by performing the epiocular eye irritation test. Finally, based on the SCCS notes of guidance, a hypothetical risk assessment case study for a day cream containing 0.1% of this extract showed that there is no safety concern as the margin of exposure calculation based on read-across approach from fulvic acid are satisfactory. To conclude, the results demonstrated that these microbial natural products can be considered as potential candidates for industrial development that can have successful applications in the cosmetic industry.